on the outside of the cell, these glycoproteins serving as surface receptors, things that bind to them, then may be brought into the cell, may affect the cell in some way.
You call them glycoproteins?
Glycoproteins? Glycoproteins, can you see that?
No.
Glycoprotein. Glyco for glycogen or sugar group, protein. So it's a combination of a sugar and a protein. We also have glycolipids that will study this as well, a sugar and a fat that are on the surface. These serve as identifiers so that other cells can recognize what they are. They serve as receptors so you can have cell-cell communication. And of course, it's these receptors that bacteria and viruses take advantage of when they infect cells. Those receptors are not there so that they can be infected, so that the virus or a bacterium can infect them. They're there for another purpose. And the virus or the bacterium just happens to have a molecule that fits into that receptor and then allows it to infect the cell. Movement across the membrane, again, a little bit different than what we see with the prokaryotes. Diffusion of molecules is a means by which things can move into the cell. Osmosis is another means. And we see with eukaryotes that diffusion is used more frequently than maybe in prokaryotes. For instance, you can have a high blood sugar content circulating that would be greater than the blood sugar content in the cell. And so glucose then would diffuse into the cell without the need of energy. So we do see diffusion as a process that the eukaryotic cell uses more frequently than a prokaryotic cell would use. But the main way that that material gets into the cell is by a process called endocytosis. So endocytosis is a way of engulfing, surrounding the material with the cell membrane and bringing it into the cell. Now there are two types of endocytosis. Pinocytosis is a means by which the cell drinks, if you will, brings in liquids. Imagine that you've got surface receptors on the cell here, and so little molecules of fluid might adhere to these surface receptors, and then what happens is that the membrane pinches in so that you end up with a vesicle now, membrane bound with the fluid that had initially attached to the surface then. So in this way, little fluid and stuff can be brought into the cell. It's not the main way of bringing in big debris and stuff like that, but it is a way of bringing in small particles. Mainly you could think of it as drinking, how the organism drinks. Compare that to phagocytosis. Phagocytosis is a little bit different in that, again, you're going to have things that are adhering to the surface of the cell, but in this case, you get an extension of the of the cell membrane like a pseudopod, like pseudopodia, and then they extend out and surround the material, creating a little vesicle, bringing it into the cell. In both cases, there'll be other vesicles in the cell filled with digestive enzymes. These are lysosomes. And lysosomes will then fuse with the bodies, the vesicles that have been brought in, and degrade the material that's there. So this is referred to as an endosome when we've brought in fluids. And this is called a phagosome when you've brought in bacteria and other larger debris. And this is a lysosome, and it is filled with digestive enzymes that will break down the material that has been brought in. And then, once everything's been digested, the cell wants to get rid of the material, and it uses a process called exocytosis to get rid of the waste material. In that example, what happens with exocytosis, imagine now that this lysosome fused with this phagosome, the material was broken down. What could be used, what could be absorbed by the cell was, and then this makes its way to the membrane, fuses with it, and releases the material to the outside of the cell. So endocytosis is the main means by which eukaryotic cells are able to move things across the membrane. Of course, they still have the protein channels and the protein carriers that prokaryotic cells do, and those are certainly used to move material into the cell. Now let's go back to the structure of the eukaryotic cell. Because eukaryotic cells don't have a cell wall, they have a cytoskeleton that helps provide the cell with its shape, give it its integrity, if you will, its structure, if you will. And there are three main tubules that make up the cytoskeleton. These tubules here, they serve to provide the cell with its framework. We have microtubules. And microtubules are used in the formation of mitotic spindles. So when the cell goes to divide, microtubules are responsible for splitting the DNA apart. Also, the movement of organelles through the cell. So mitochondria or chloroplasts, other organelles, they move along the microtubules that are running through the cell. So you'll have these long, hollow tubes that serve to run through the cell. They can be broken down and manipulated as needed, but the organelles and stuff are able to move along those microtubules. In addition to that, we have actin filaments. These actin filaments, they play a role in the formation of pseudopods. They can polarize and depolarize in the formation of the pseudopods as the cytoplasmic streaming takes place. And then finally, you'll have little microfiber, intermediate fibers that are running through the cell, kind of like rope, if you will. Again, just to add structure and framework to the cell. Now, eukaryotic cells also can come with a flagellum, but this flagellum is going to be very different in structure than the flagellum that we see with prokaryotes. This flagellum here is made from a product, or from a molecule called flagellin that again is unique to bacteria. Hopefully you're starting to catch on to this thing here that we've identified peptidoglycan that's unique to bacteria. We've identified lipopolysaccharides that are unique to bacteria. Now we've identified flagellin, which is unique to bacteria. Think about the drugs that we create and use. If you're going to use a drug that is going to be effective against bacteria, but not hurt your own cells, isn't it good to have a drug that's going to target something that only bacteria have? So any drug that targets flagellin, lipopolysaccharide, peptidoglycan, we can give without necessarily having fear that it will affect our own cells.
It's just flagellum.
Flagellin. Yeah. Now, in addition to having one flagellum, some microbes, some eukaryotic cells have cilia, and we've talked about this in lab. Structurally, cilia are the same as flagella. The difference is that they're short instead of long, and they come in thousands instead of one or a couple. Our eukaryotic cell is going to have a nucleus, and inside the nucleus we'll have our genetic material. Now our nucleus has two membranes. Surrounded by two membranes, and it has pores that allow for things to move in and out of the nucleus. Additionally, we'll note that our DNA is now linear. Prokaryotic DNA, circular.